Hello students, welcome back to our YouTube channel Zoologic Minds. In previous class, we studied about the general characteristic features of the phylum Cnidaria. If you have not watched the video, you can get link of that video from description box. And today we are going to learn about the defense mechanism in Cnidarians, which is the characteristic feature of the coelentrates. Let's get into the topic. First, we should know the concept of nematocyst and cnidoblast. So, nematocyst, this is the defensive structure which is present in the walls of the cnidarians. Now, in this diagram, we can see that this is epidermis layer, this is gastrodermis, and in between these two layers there is mesoglia. In the epidermis layer, there are interstitial cells which are modified into cnidoblast cells. And nematocysts are not cells, they are actually organelles which are present in the cnidoblast. So, due to the presence of cnidocytes in the epidermal layer of the cnidarians, they are known as the cnidaria due to the presence of cnidocytes. Now, cnidocytes helps in defense, but they also helps in the locomotion, adhesion, and capture of the prey. These, as I told you earlier, these cells are organelles, not actually cells. They are organelles which are present in the cnidoblast. They are modified from the interstitial cells of the epidermis. They are not found in gastrodermis. This point needs to be remarked because most of the students get confused that the cnidoblasts are present in the gastrodermis, but not. It is present in the epidermis only. Now, when they are fully developed, they migrate to the tentacles by the amoeboid movement. Now, the structure of cnidoblast. The cnidoblast is an oval, rounded, with a basal nucleus. This is the structure of the cnidoblast, and inside this the nematocyst is present. This is the nematocyst, and this whole cell is the cnidoblast. It is oval or rounded and with the basal nucleus. This nucleus is present in the base and it is shifted on one side, either on left or right. Okay. And inside the cnidoblast, an oval pyriform sac is present, which is called as stinging capsule. So this is the oval pyriform bladder, this one, and this is called as the stinging capsule. It is made up of chitin. Now the bulb is filled with poisonous fluid which is called as hypnotoxin. It is chemically the mixture of proteins and phenols. One end of this bulb is extended as narrow, long, hollow tube-like filament which is coiled around the poisonous sac. This filament is called as thread. So this is the bulb and this is one end of the bulb which is extended narrow. Now this part is getting narrowed gradually. Okay, it is which in a rest condition it coils around this bulb poisonous sac. It is called as thread tube. The base of this thread tube is swollen to form a shaft. Now this swollen part of the thread tube is shaft. This shaft contains barbs and barbules. These spines, which are large, are called barbs or stylets, and these smaller spines are called as barbules, and this structure is covered by operculum. The outer end of the cnidoblast projects freely beyond the epidermal surface as a tiny pointed hair-like structure called cnidocyl or trigger. Now, this is the cnidoblast, and it, it is freely projected beyond the epidermal surface by this cnidocyl. It is also called as trigger. It has 9+2 arrangement pattern, as we can see in cilia. There is also 9+2 arrangement, and here also 9+2 arrangement is present. Now the cytoplasm of the cnidoblast contains contractile fibrils. Okay, and there is some restraining thread also called as lasso. So this is the restraining thread which is called as lasso. It is present at the base of the cnidoblast, and it protects the nematocyst from being thrown out of it. Okay, now this explains the defense mechanism of the cnidarians. Okay, the discharge or explosion of the nematocyte takes place when the cnidocyl is stimulated by the food, prey, or enemy. So you need to mark this point that for the discharge or explosion, both mechanical and chemical stimulation are necessary. Okay, the wall of the nematocyst remains impermeable to the water except during the discharge. Okay, so most of the time the wall of the nematocyst is impermeable to water, but when it gets mechanical and chemical stimulation, the discharge or explosion of nematocyst occurs, during which the nematocyst becomes permeable to water. And due to increase in permeability, the osmotic pressure inside the capsule increases. And due to increase in the pressure, the whole nematocyst explodes outside, and the thread tube everts, which now the base of the thread tube has a shaft which contains barbs and barbules. These barbules get anchored to the prey and causes death by releasing some toxins, and that's how the defense mechanism of cnidarians work. Now you need to note the point that once the cnidoblast is used, it gets replaced by another cnidoblast within 48 hours, approximately 48 hours, and it cannot be used again. It is discarded. So do you remember in previous class I told you that the cnidarians exist in two forms, either polyp or medusa form. So we need to understand that what is the difference between the polyp and medusa form. The polyps are sessile and they are attached to substratum. However, the medusae are free swimming and they are umbrella shaped, and the polyps are cylindrical. Now movement, obviously polyps can't move because they are sessile, so they are sedentary animals. However, medusa are free swimming, that's why they are mobile. Tentacle orientation, in polyps the tentacles are upwards, but in medusae the tentacles are downwards. Mouth position, in polyps the mouth position is upward, but in medusae it is downward facing. Reproduction, polyps produce asexually but medusae produces sexually. nervous system. It is concentrated at the base. However, the nerve nets are spread across the umbrella in medusae. Polyps are typically smaller in size, but medusae are conspicuous, means they are more large and visible. Polyps are less sensitive to the change in environment. They are sensitive to the stimuli and they respond to the environment very sensitively. In next class, we are going to discuss about the general characteristic features of phylum Ctenophora. And if you like my video, subscribe to my channel, press bell icon so that you can get notified about the latest video on my channel. Thank you for watching my video. In next class, we are going to discuss about the general characteristic features of phylum Ctenophora. And if you like my video, subscribe to my channel, press bell icon so that you can get notified about the latest video on my channel. Thank you for watching my video.